going to show my screen. Can you see it now? Okay, yeah. So thank you, Petter. As you already mentioned, I'm a student and I'm currently enrolled to the master's program Fish Biology at Humboldt University in Berlin. But I conducted my bachelor thesis at Linnaeus University in Kalmar last spring, and this is what I'm going to present today. So I will also address the stickleback-pike interactions, and I will focus on the question if olfactory cues inform sticklebacks on whether pike is a predator or prey. So as you already heard, the coastal fish community has drastically changed among large parts of the Baltic Sea, including pronounced declines of the top predator northern pike in many areas. And in the graph, you can see the trends in abundance of pike and free-spine stickleback in the Kalmar Sound area. And if you first have a look at the black line, you can see that the abundance of pike collapsed between 1995 and 2000. And as you already heard, a lot of potential reasons have been linked to this decline, among that habitat degradation, fishing, pollution, seal and cormorant predation, or eutrophication. And even though there have been a bunch of conservation and management actions like closure of the pike fishery or the initiation of spawning habitat restoration, the pike populations haven't recovered until today. So if you now have a look at the blue line, you can see that around 10 years after the collapse of pike, the trends in abundance of stickleback, which is a typical prey of pike in the Baltic Sea, increased exponentially, up to 45-fold in the center Baltic Sea, potentially favored by reduced ocean pressure, by reduced competition for zooplankton, and climate change. So as you already heard, today the regime shift from former top predator-dominated ecosystems to stickleback-dominated ecosystems. And a factor that is reinforcing this regime shift and impairing the capacity of pike populations to recover is the stickleback predation on pike juveniles. So Baltic pike use two different reproduction strategies. As you already know, they spawn either in shallow brackish water or in adjacent coastal freshwater, usually in wetlands that they reach by migrating upstream smaller streams or ditches. And these wetlands are suitable spawning grounds for these freshwater spawning pike, so-called Anadromous pike, because they provide dense vegetation. They provide high zooplankton concentrations in early spring, and very importantly, due to rivulets or fishways at the outflow of the wetlands, they are not accessible to species with lesser swimming ability, for example, stickleback. The recruitment period of pike coincides with the timing of stickleback migrating from their offshore pelagic habitat into coastal areas for spawning. And some of these sticklebacks form dense aggregations outside the wetlands used for pike spawning and even within the streams that connect the wetland to the Baltic Sea. And very interestingly, and that is very important, that in some locations, the stickleback arrive in the streams even before the pike larvae started emigrating from the pike, from the wetland. So that means if the stickleback aggregate outside these wetlands, because they are waiting for the pike larvae to leave the wetland, they are able to localize these spawning grounds without visually detecting the pike larvae. And whether the prey, the emigrating pike larvae end up as stickleback food or not largely depends on their body size and their body depth. And this graph is based on the total length and gape limitation measurements from sticklebacks that were sampled in the Kalmar Sound area again. So on the y-axis, you can see the total length of pike from 0 to 180 millimeters, and on the x-axis and on the y-axis, you can see the proportion of stickleback. And when the pike juveniles are still very small, the y-axis shows the proportion of sticklebacks that are able to prey upon the pike larvae based on their gape size limitations. But with increasing total length of the pike, the y-axis shows the proportion of stickleback that fit into the pike's mouth. So the relationship of the stickleback gape limitation and pike body depth predicts that the critical size for pike to outgrow the predation window of stickleback is approximately 33 millimeters. And after outgrowing this predation window, the pike are neither prey nor predator until their gape size limitations enable them to predate upon the smaller size classes of adult stickleback. And all stickleback are potential prey to pike that reach 175 millimeters. So, summary, the background: sticklebacks predate on juvenile pike, but they are also preyed upon by adult pike. Sticklebacks somehow localize the wetlands used by Anadromous pike for spawning without visually detecting pike larvae, and they aggregate outside these wetlands, even though spawning adult pike potential predators inhabit the wetlands. So the aim of this study was to investigate the role of chemical communication in this predator-prey role reversal, and therefore to disentangle olfactory cues and to find out what kind of information they give. And therefore we developed this experimental arena that was divided into 3 interconnected chambers. And by simultaneously adding water from 2 different sources, which is indicated by the 2 different colors, the water and the chemical information that is associated with this water mixes within the outer chamber and then slowly pushes into the middle chamber. And in the middle chamber, the water leaves the system through an outflow in the bottom. And this creates a unidirectional flow of water that prevents chemical cues from one outer chamber to enter the other outer chamber. And the stickleback is then placed in the middle chamber in an acclimatization cylinder, and as soon as this cylinder is lifted, the stickleback could choose between both outer chambers based on the chemical information in the middle chamber. And to test whether this design was actually working, trials of colored water were conducted. So we hypothesized that stickleback need additional chemical information to distinguish between adult and juvenile pike odor. And to test that, the stickleback was subject to 1 out of 6 different treatments. And in each treatment, the stickleback could choose between the control chamber containing only brackish water and the cue chamber containing different kind of cues. So in the first treatment, the stickleback could choose between the control chamber and the cue chamber containing pike larvae odor. In the second treatment, the stickleback could choose between the control chamber and the cue chamber containing, again, pike larvae odor, but this time in combination with diet cues from digested zooplankton. In the third treatment, the cue chamber contained adult pike odor, and in the fourth treatment, the cue chamber contained stickleback alarm pheromones that are also referred to as Schreckstoff that were obtained from homogenized sticklebacks. And maybe I should explain that a little bit more in detail. These stickleback alarm pheromones are localized in the tissue of sticklebacks, and they are released by mechanical damage, for example, during a predation event. And these substances that are released to the water then can, for example, be used by conspecifics for assessing the predation risk. In the fifth treatment, the cue chamber contained adult pike odor together with Schreckstoff, and thereby representing the chemical information that is released by a pike that has predated on sticklebacks before. And lastly, in the no cue control treatment, brackish water was tested against brackish water. So to test whether the sticklebacks preferred the cue chamber over the control chamber in the different treatments, the time the stickleback spent in the cue chamber was compared to the time it spent in the control chamber. And this was based on video recordings of 120 trials, each of it 5 minutes long. The data was then transformed binary to quantify the preference, and then the data for the cue chamber preference, or for the cue chamber, so whether it was preference or not, yes or no, was used to analyze the effect of the treatment on the cue chamber using a generalized dynamics model. And in the end, 96 trials were included into the analysis because in 24 of the 120 trials, the sticklebacks didn't choose one of the outer chamber, and therefore they were excluded. So in the graph, you can see the 6 different treatments on the x-axis and the Q chamber choice in percentage on the y-axis. And first of all, there was no general effect of the treatment on the Q chamber choice. On the factor level, however, differences between the treatments could be observed. And in the control treatment, which is on the very left side, the sticklebacks preferred the assigned Q chamber in 9 out of 18 times, and that resulted in a Q chamber preference of exactly 50%, and therefore the control treatment was used as an intercept to pairwise compare the other treatments. And in the larvae treatments, the larvae treatments had no effect on the Q chamber choice, regardless on whether direct Qs were present or not. Adult Pike odor and Schreckstoff did not have a significant effect on the Q chamber choice when they were tested alone, but when they were tested in combination, the sticklebacks avoided the Q chamber significantly. So there was no general effect on the treatments of the Q chamber choice, and this suggests that chemical communication is of rather small importance for offshore living. For these offshore living pelagic sticklebacks. And this seems likely since stickleback reliance on olfaction is usually elevated when vision is reduced, for example, in turbid environments. However, the differences in Q chamber choice between the treatments suggest a general use of olfaction in the tested population. So there was no preference of the Q chamber in both larvae treatments, and this suggests that the tested stickleback were not attracted to the larvae odor. And in case of the stickleback aggregation outside the wetlands used for Anadromous pike spawning, this suggests that chemical detection of pike larvae might play a minor role in locating these wetlands. And this further means that pike recruitment might not be the major reason for stickleback aggregations outside these wetlands. So the sticklebacks did not avoid the Q chamber containing only adult pike odor, which means that they didn't associate the pure pike odor with predation risk. And even though there was a clear trend towards avoidance of the Q chamber in the Schreckstoff treatment, the sticklebacks only avoided the Q chamber significantly when adult pike odor was combined with Schreckstoff. And this is in support of our prediction that sticklebacks need additional chemical information to chemically label a pike as a predator. Applying these results to the predator-prey interactions of sticklebacks and Anadromous pike, it explains why stickleback do not avoid the wetlands that are inhabited by adult pike potential predators. So considering that sticklebacks aren't able to enter the wetland, the adult pike cannot predate upon sticklebacks in the wetland. And that means that no stickleback Schreckstoff is draining out of the wetland. And now taking into account that the avoidance of the tested sticklebacks in this experiment was only observed when combining the adult pike odor together with Schreckstoff, we assume that the sticklebacks outside the wetland simply do not care about the adult pike odor that's draining out of the wetland. And to aid management regarding the conservation of Anadromous pike in the Baltic Sea, these results suggest that adding stickleback alarm pheromones to heavily affected streams might prevent this stickleback aggregation. Whether this is an effective tool or not needs to be investigated thoroughly in further studies.